For today's dissection, we're looking at the digestive tract of a small pony, likely to be an Exmoor or a Dartmoor pony, going by the size of the digestive tract. Obviously, if we had been looking at the digestive tract of a larger horse, such as a 16-hand thoroughbred, for example, the digestive tract would have been bigger compared to what we're seeing today. So we've got everything here. We've got the stomach with the spleen attached to it as well. So the spleen in the horse is responsible for red blood cell storage for when the horse requires it. And as you can see, it is very closely connected to the horse's stomach. From the stomach, we then go into the small intestine, and you can see here how that the small intestine is intermingled with the large intestine, and particularly the small colon aspect for that. Now, in the small intestine, we've got a lot of digestion and absorption taking place, particularly of starches, proteins, and fats. And when we're looking at the absorption of those sugars, of fats, and of protein, it means we need to make sure there's a good blood supply going to the small intestine. And you can see here within the connective tissue how the different blood vessels that are moving towards and away from the intestinal tract. So the stronger the blood supply is within these regions, and there's a nice bit coming up just here now, the stronger the blood supply, the more absorption is likely to be taking place in that section. So within the small intestine of the horse, we're looking at the absorption of sugars, of fatty acids, of amino acids, so where protein has been digested, before we get to the large intestine. So the horse's large intestine consists of the cecum, the large colon, the small colon, and then finally the rectum and the anus. And what you can see here on the screen is what we call a flexure. So the flexure is where the large colon, in this case, really bends back onto itself. And these flexures are often the location of impactions that result in colic. So that could be impaction of food, it could be impaction of gas, it could be as a result of worm infestation as well. But those are normally the areas where colic, particularly impaction colic, can take place. And there are a few of those flexures within the large colon of the horse as we go through. So we've got the cecum in view just there as well. So the cecum is a blind sac, if you like, where we find a lot of the microbes and where a lot of fermentation takes place. Moving on then to the last part of the digestive tract, at the top you can see the last part of the small colon, and we then get towards the rectum and the anus, which we've got. Now, with this specimen that we have on the table in front of us, we also have the bladder, which is just being considered further. So we have got an empty bladder in this case, luckily. But you can see how the bladder is closely connected to the anus there at the end. So that's just given us a quick overview of the horse's digestive system. So we're looking here at the horse's stomach with the spleen attached to it. The spleen is being kept in place with very strong connective tissue. As you'll be able to see in a moment, you can see how we've got blood supply going through, a bit of blood supply going through there. But that connective tissue is really strong. Although it doesn't look that way, it does keep it all in place. So the horse's stomach has got generally what can be described as a J shape, where we've got the entrance and the exit located very closely together. We've got the esophagus located there. We've got the great curvature, so the larger curving side that was just on the outside, and where we're going to be making the incision round, which basically goes from the saccocecus area, so the top part of the stomach, all the way through to the pyloric region, where we would find the pyloric sphincter. As you can see, so we've got slowly got the stomach being opened up, and we'll be able to get a nice view of the glandular region and the non-glandular region as well. So you can see some of the mucus that sits on top of the horse's of the feed that is located within the stomach. So this horse has had a high forage diet, as we can see from what we're finding there. And what we're also seeing on the right-hand side of your screen there, the little red worms are basically bot larvae. And bot larvae generally will, when they do infest the horse, they do have a tendency of locating themselves in the squamous region of the stomach, so the non-glandular region of the stomach, where they can hook themselves nicely into the lining. So keeping in mind the lining of the squamous region of that non-glandular section is thicker compared to that of the glandular section. Also, obviously, within the glandular section, we also find more acid, so it's less of a hospitable and welcoming environment for those bot larvae. We had another worm just there as well, so that does promise something possibly for later on in today's dissection. We may see some more worms, and therefore it's very likely indeed that we're looking here at a feral horse as well, a free-ranging horse. So having had the food removed from the stomach, you can see we've got the glandular region at the bottom end. Just wait for the hands to be moved away and the food to be taken away from that region. Now, normally speaking, we would find the glandular region of the stomach being a bit more purpley reddish in color. This one does look slightly pale, which might be the result of the worm infestation that this horse has had as well. So that's just something to be aware of. Normally, as I said, we would find a nice purpley red color as a result of the hydrochloric acid that is being released in that particular section. So what we can see is we've got the bot larvae, as I said, that sit nicely in the squamous region of the stomach, and they really hook themselves into the squamous lining, as we'll be able to see in a moment. So you can see there where we've got the glandular region of the stomach. You can see how it is that bit more slimy in terms of its structure compared to the squamous region. So where we're now getting some of those bot larvae out of the squamous region, you can see the damage that they do. So they hook themselves into the squamous region of the horse's stomach. Nice thick area, but you can see what damage they leave behind at that point where they hook in. Now, obviously, within the stomach, we have got two sphincters. We've got our cardiac sphincter and we've got the pyloric sphincter. So the pyloric sphincter is the area being pointed out just now. So that's where we move from the stomach into the small intestine, and moving particularly into the duodenum part of the small intestine. Whereas with the cardiac sphincter, which is being stoked out now, that is where the esophagus enters the horse's stomach. And the cardiac sphincter in the horse's stomach is so tight as a muscle that is the reason why the horse is unable to regurgitate its food. In contrast to that, looking at the pyloric sphincter, that's where we're seeing a much softer muscle, which makes food easier to pass through. Again, around the stomach, you can see that connective tissue that is helping to keep the stomach in place within the horse's abdominal cavity. From the stomach through the pyloric sphincter into the horse's small intestine. So on average, the horse's small intestine is about 22 meters in length and is made up out of the duodenum, the jejunum, and the ileum. To keep everything in place, again, along the small intestine, we find a lot of that connective tissue with a very strong blood supply, allowing for absorption of nutrients from the small intestine. And what you can see here is that there's an attachment point, if you like, where that connective tissue all comes to one point and make sure that the small intestine stays in place. And that's to prevent any mixing up and any recoiling of the small intestine within the abdominal cavity that could result in blockages, for example. Now that connective tissue, as we just saw, and we're going to be seeing that a bit more in a moment, has got a very strong blood supply throughout it. And that blood supply, as I mentioned, is needed to allow for absorption. So within the small intestine, we see starches and sugars being digested, fats being digested, and proteins being digested. And that all happens using chemical digestion, so through the use of enzymes. Once those products have been broken down, they then are able to get absorbed into the horse's bloodstream through the intestinal wall. And that's where that blood supply is so important, to make sure those nutrients make it through into the horse's body so the horse can use it. Now, within that connective tissue as well, you can see those black nodes located, and those black nodes are lymphatic tissue. And as we know, lymphatic tissue will sit very closely alongside the horse's normal blood, alongside the blood supply itself. Now, although that connective tissue might seem very thin and very weak, it is actually very strong, very resilient, so it won't break easily at all. So we've spread the small intestine out here. So it was just pointed out there was the ileum part, and on the front side we've got the duodenum. So we've got the duodenum, the jejunum, and the ileum. And we're going to have a look at the different contents and the difference in wall structure here as well. So starting off looking at the duodenum portion of the small intestine, you can see we've got quite a bit of gooey substance there, and obviously it's important to keep in mind that within that duodenum we've got the release of pancreatic juice and bile through the sphincter of Oddi, and we've got the release of secretions from the small intestinal wall. And those secretions, and the pancreatic juice, the bile, and the secretions coming from the small intestinal wall, all contain important substances to allow to start the breakdown of food in the small intestine. So it's where we find enzymes such as lipase and amylase, for example, that can start off the digestion of fats and starch, amongst others. Moving on then, we go into the jejunum part of the small intestine, and here you can see that we've again got a different composition of the food. It's become a bit thicker, so we've had absorption already starting to take place in this area, and you can also see the wall of the jejunum section being that bit different. So keeping in mind that within the small intestine we have the inner wall is made up out of villi and microvilli to increase the surface area. So increasing the surface area will allow for more opportunity for mixing of food with enzymes, but also to increase that absorption content as well. So we've got a more ribbed structure there in the jejunum already compared to the duodenum. Moving through then into the ileum, so the last part of the small intestine, we can see it's a lot more watery at this point. So most of any materials that could be digested here will have already been digested. And you can see that the wall here is also a little bit more translucent almost. So we're just going, hopefully this is coming across as well on the cameras as we'd hoped, but just a bit of comparison of those three sections. So where we've got the duodenum, the jejunum and the ileum. And you can see you've not only seen how the different structures are different from each other, particularly in terms of the lining, in terms of the wall, but also thinking about the content. Now, chances are this horse mainly was receiving forage, so would not have had as much digestion, as much food being digested in the small intestine as expected. So continuing to make our way now along the small intestine, because all of this that you're seeing at the moment is all still part of the small intestine, so particularly looking at that ileum being expanded onwards. And we will soon be putting it all alongside each other, so you get an even better idea of the length of the small intestine. So having pulled all of it apart, you can see the stomach there at the top of your screen, and then moving to the right from the stomach, we've got the duodenum that then starts folding back on itself, carrying on before we move into the jejunum section of the small intestine, and finally the ileum section. So this gives you quite a good indication about the overall length of that small intestine that was initially all crimpled up, held together with that connective tissue, and where we had those various attachment points as well. So as a reminder, we're looking at the overall length of the small intestine on average being about 22 meters. Now over there we've got the cecum, and we've got the ileum already joining there with at the ileocecal junction. So here you can see the ileum as it connects to the cecum itself. So we've got the cecum on the right-hand side here, and we'll separate that out in a moment so you can get a better look. Now the cecum has got quite a strange design. It's best described as being a comma-shaped blind sac, which is full up with microbes, because this is where fermentation of fibers already start to take place. And you can see that along the wall it's quite folded. It's got a lot of folds there, and we've got a thick fibrous cord running all down the cecum to keep everything attached together as well. In terms of its design, the cecum is a bit odd in that where we find ileocecal junction, close, almost next to there, we will then find the entrance to the large colon as well. Now, where we're currently cutting this open, the food is still slightly frozen, so it will look a little bit more solid than that we would normally expect. So within the cecum, as I mentioned, this is where we find a lot of microbes living, particularly fibrolytic and amylolytic microbes that are responsible for the fermentation of fiber or forages. So any grass-based products won't really get digested until they make it into the cecum. And they can sit in the cecum for quite some time. They very much sit, it sits there and brews. It sits there and ferments. So the microbes make the most out of the forage sitting there, and it can stay in the cecum for up to 7 hours. Now, even on the inside where we've removed the food, you can see that highly folded lining as well, and how it's all ribbled up together. So again, increasing that surface area, allowing microbes to move their way around, and allowing for an increased capacity. There you see this ileocecal junction just at the top of the screen being shown, kind of just missing out on that. But then as I said, right next to there, we then have the entrance to the large colon, so the next section of the large intestine. So within the large colon, which we're moving into, the large colon and large intestine generally can get used interchangeably, but I tend to refer to it as the large colon to make it clear that with the large intestine, we're also including the cecum, the small colon, and the anus and the rectum at the end as well. So the large colon itself is normally about 3.5 meters in length and has got quite a large capacity. It folds back on itself, as we saw earlier on in the video, and we'll get a nice view of that now as well. So you can see how the large colon moves from the top of your screen down to the bottom, where we've got a flexure. So the flexure is where it coils back onto itself and then makes its way all the way back up. So all of that is the large colon. Okay, so you can see again we've got a number of connective tissue that helps to keep it all together as well. And we still see a ribbed structure in the similar ways that we saw in the cecum earlier. So starting to have a look then at some of the food material within the different parts of the large colon. So we're seeing here more that fibrous material. So we've had the grass and the hay that this horse has been eating, that has been sitting within this, has moved from the cecum down into the large colon. And in the large colon, we still have microbial fermentation taking place, as well as absorption of those volatile fatty acids that get produced, and byproducts such as amino acids and water that will get absorbed here, that will start to get absorbed here as well. Now, along the large intestinal, the large colonic wall, you will also still find that fibrous attachment, that fibrous cord that keeps it all together and allows for that ribbed structure that you're seeing here. So I'm just moving, having a further look inside of this. Once we open it up, you'll be able to see that we still actually find that ribbed structure within that food material as well. So you can see those different bulges present there. Now, as we move through the large colon, we'll start to see this food material getting drier as we go on. But this starts to already look more as feces as we expect it to be. So the flexure that you're seeing here, this is where what we refer to as a tight coil up of a tight bend in the large colon, and it is an area where colic is most likely to occur as a result of food matter getting blocked, but also it's a location where worms are likely to actually insert themselves and cause blockages as a result of that. So moving towards some of the later sections of the large colon, we did find some red worms in there as well. But one of the main things here really is to look at the difference in the large colonic wall. So we're seeing the wall here that has become darker in color compared to that earlier section that we looked at. And it's also got a bit thicker in structure as well, and thicker to the feel, to touch. So this is where we would start to see some more absorption start taking place already. And certainly when we start to look at that last part of the large colon, that was just a bit of frozen food. But starting to compare what we've got at the end compared to the beginning, we can start to see that that matter has started to dry up. So within the large colon, within the large intestine overall, we do find a lot of water absorption. And sometimes the horse's large intestine as a whole can also be referred to as a water reservoir. So the last section then, we've got gone from the large colon into the small colon, making our way all the way around until we eventually get to the rectum and then the anus there. Now, alongside the anus, we also find the bladder. Because of its location, this would suggest that we're looking at the digestive tract of a filly. If it was a stallion or gelding, the bladder would have been located further away here. So where we were looking at the fecal material that we found in the large colon, let's have a look at the same when we're moving into the small colon. So what we can see here is that it's all starting to already look that bit drier as well. So we've still got more water absorption taking place within the small colon. And it's important to keep in mind that depending on what kind of forage we're feeding our horses, they will also be absorbing B vitamins within the large intestine overall, as well as water and amino acids that are produced as a result of microbial fermentation. Now, as you can see, we've got the small colon still has that same ridged view that we had, same as with the large colon, but you can see how the matter is becoming drier as we move through. So that would suggest some good water absorption. The wall lining here that you're seeing, you can see how it's quite thin. As you can see, the finger moving underneath it very, very clearly. So it gives you an idea about how thin the wall of the small colon actually is. As we make our way around through there, we're still finding some red worms in here as well. But you can again see how it's getting drier and drier. And we're really starting to get those fecal balls starting to form that we normally expect to see when the horse does do a poo. Now, those balls are being formed as a result of muscle contractions throughout the small colon. So same throughout the entire digestive tract, you would have peristalsis taking place here, and we have another process taking place called segmentation. So coming to the end of the small colon, you can really start to see those fecal balls being formed as a result of those muscle contractions. And they would then make their way and pass through the rectum and the anus. So looking at the rectum there, you've got a bit of there where it's already coming out, but you can see that the rectum is thicker compared to the earlier structure, the earlier bits of the small colon that we saw. And that's because the rectum, we're starting to see more muscle being present within the wall of the rectum. Because when we get to so as the food gets moved down the small colon into the rectum, that's